Bioprinting is a cutting-edge technology that helps to create a specialized artificial human tissue to replace it in the human body. And this is very patient-specific technology, and this technology can build really complex structures with the help of bioinks, for example.
Bioprinting is a new discipline in what is called additive manufacturing, that uses biomaterials called bioinks to print on a 3D scale scaffolds mixed with cells. This is where the bio term comes into place now. What we're trying to do with bioprinting is basically to reproduce natural tissues, natural living structures.
Bioprinting is a tremendous technology. It's a form of additive manufacturing which allows users to take cells and a material known as a bioink to create a shape that is of physiological relevance. And it's this control over the geometry as well as the cell positioning that enables users to really capture in vivo environments. And so through the power of bioprinting, you have the ability to create an environment that so closely mimics the natural body that truly, I think it will unlock the future of healthcare. A bioink is a material. Its function is simple, yet critical. A bioink is the material that allows for the printing process to take place. So in the example of extrusion printing, the bioink's first job is to ensure that the printing process can take place. And what that means is that the cells can pass through a nozzle unharmed and retain their function. The second part of a bioink is to ensure that the material allows the shape to remain. So the desired printed construct or the geometry needs to remain after the printing process. And the third, and perhaps the most important part of the bioink, is that it should be an environment that caters to the cell. It should allow and enable the behavior that the researcher or the user wants. And so picking a bioink is extremely important because it's what allows the biology to take place.
Our tissues are based in cells and materials. Now, this is how we build our bodies. So basically what we do is mimic this with biologically compatible materials, typically either materials that we take from nature or that we synthesize artificially to mimic these nature existing materials, and then we build 3D structures with that using also cells. There's the bio term here now, cells with the materials, and then we reproduce with these organs, tissues, regenerative materials.
Bioinks consist of specific materials. We in our project uses gelatin-based materials or collagen-based materials, and these materials help to protect cells which are also in the bioink, and these growth factors to be delivered at to the right point at the right time. The selection process is done by what is already in your body. What do you have for surroundings? And the human body consists of gelatin and collagen, and this is very a natural material in this case, especially for ligaments, cartilage, and this kind of materials we are looking into.
Selecting a bioink is somewhat of an art. It requires an in-depth understanding of your target tissue, the cells that you are working with, the type of construct that you're going to be printing out. But ultimately, it comes down to picking a material that best represents that part of the human body. A material that will allow the cells to behave in a manner that is as close to the in vivo environment.
So what you need in order for a biomaterial to be a good bioink are three things. First, you need the material to be compatible with cells. And this you can get either, like we do in the TriAnkle project, by taking nature existing materials. In the TriAnkle project, we use collagen, which is our main protein in the body that gives us structure, or gelatin, which is derived from collagen. So either you take nature derived materials, or you take materials that mimic natural materials. And in any case, these need to be materials where cells are happy to be around. So cells need to be need to live well surrounded by these materials. Now, this is the first thing you need. Second thing you need is the material to be flowable. So you need a material that can flow homogeneously so that you can print and generate defined structures with the material. Very important. Otherwise, if you don't have that, you would make drops and you would not generate structures, defined structures. And the third property you need is the material to be able to strengthen after printing. So in all the printing processes, first you deposit the structures, so you make the forms, and then you strengthen somehow. And this strengthening process is the third quality you need in a bioink. This is typically obtained or generated by physical strengthening processes like curing with radiation or chemically, or with pH or ion adjustments in the medium. So in the TriAnkle project, we are in a very fascinating phase of the project now. We have already developed all the tools that we want to use in the project. We have developed our bioinks based on collagen and gelatin. We have developed our stem cells. We have developed growth factors protected so that we can now combine all these materials together and test these new therapeutic options we want to develop for tendon cartilage repair. And now we are starting in different levels of in vitro and soon also in vivo testing. We are starting to test the efficacy and the use of the solutions that we have thought of at the beginning of the TriAnkle project.
We always use the most appropriate animal models in our studies in research. In the case of TriAnkle project, we are using the sheep animal model. The sheep is a large animal model, is very similar to the humans in size, and also the physiology and the anatomy of the animal is very close, very similar to the human situation. So it makes the sheep an excellent model to analyze the evolution of the pathology and also for the testing of the new treatments developed in TriAnkle project. In TriAnkle, in the animal studies, we are using two different approaches. One of them is clinical imaging, which is a non-invasive technology, also used in human clinics in the hospitals. So we are using the same technologies in the animal models that in the human patients. This clinical imaging permits the evaluation, the monitoring of the pathology and the therapeutic treatments in the animals along the time. So we can have a lot of information during the evolution of the disease and tissue healing. And on the other side, at the end of the studies, we can also we're also using histology to evaluate the microscopic outcomes in the tissues. After the implantation of the 3D implants generated in TriAnkle, we can determine the tissue regeneration, the beneficial effect of this implantation of these implants in the tissues, evaluating the cartilage regeneration, the tendon degeneration, the articular diseases recovery. So this is very important to go in parallel with these two strong therapeutic analysis.
Well, future is always something that is difficult to predict. What we hope is actually to be able to generate new tools for patients to improve their health and their recovery after injuries or after long life degradation of the tissues. And we hope that our solutions set the base for new therapeutic options for these patients. Lose the ability to walk, so bringing new solutions, bringing new ideas, new options for the doctors to treat these patients is basically our mission and our goal in the project.
As you can suppose, doctors are receiving many proposals in terms of the biological treatments in order to help our athletes. We believe that in the future we will use cell therapy because it's more power. But to use this kind of treatment, it's very important to follow the regulation, the rules, in order to have permission for use this kind of therapy. Because if everyone use this kind of therapy without permission, then maybe we can have a big problem.
We think that with this kind of therapy should be very beneficial for patients, and in our experience as a patients, they like to participate in the research, they like to participate in the clinical trials, they like also to prove the product. I mean, I think there is a lot of expectation as a patients in order to take the product as soon as possible, no, and to improve the quality of life, because the most important issue, even it's important to reduce pain, improve mobility, but the important issue is that the patient have a very good quality of life. That's very important, no?
Although that this treatment has not been approved for the treatment of this kind of diseases in patients, at the Osteoarthritis Foundation International, at the OAFI, we have developed 2 studies to see what people, patients thinks about this type of therapies. When we ask the elite athletes, they have different expectations that the osteoarthritis patients. For example, they want to recover very fast and they want to return to play as soon as possible, compared to the expectations that osteoarthritis patients have, that what they need is to have less pains in their joints and to have mobility and function again. But even that they have this difference, they were very interested in this type of therapies. They think that it will accomplish their expectations, that the tissue will recover faster, will also regenerate, and they can be good again as soon as possible.
Well, TriAnkle has many good aspects, especially for patients with problems in their knees or in their ankle, and it helps to push regenerative medicine in this area forward.
Yes, so all the degenerative-based diseases are very difficult to cure. Now, we mammals are designed to fight diseases and fight bacteria and all the microorganisms that want to attack us. We're not designed to regenerate. Now, there are other animals like reptiles that are designed to regenerate the tissues. We don't. No? Our immune system is designed to attack, not to repair. So that's why for us as humans it's very difficult to regenerate. No? One of the things that we want to do in the TriAnkle project is try to overcome this problem and try to at least generate options to go into this direction and regenerating damaged tissues.
Oh, for sure. TriAnkle is a tremendous, promising therapeutic strategy for the future for the treatment in human patients, because it's a personalized therapeutic approach. So we can generate an implant with cells or with pharmacology to treat a specific patient, a specific disease. So we can generate personalized medicine using TriAnkle with a very strong consortium and team, and also in a very advanced therapeutic strategy.
Certainly, I think the TriAnkle project is a tremendous hope for a number of reasons. First and foremost, it brings together subject matter experts from a diverse background. It takes people who really understand their disciplines and brings them together in a playing field that allows for a real development of a potential therapy. And secondly, I think the project has a very important focus from a patient perspective, working with doctors and ultimately the people suffering from the condition, and keeps that at the core, and thus creates a solution that is applicable and will really make an impact in the people suffering from these conditions.
I think the good thing about TriAnkle project is that we are different partners from different parts of the world with different expertises. So I think through this project we have learned from each other a lot. I think we have put our strengths together, always bearing in mind that the patient is the most important thing. That's why we are using different materials, different cells, different factors, and also animal models with the objective of having the best that we can make together.